Looks like people are coming in. Hi, everybody. Welcome. Thanks for joining us today for the smart skin and amazing brains of the octopus. I don't know where you are and what your weather's like, but here in my neck of the woods in the east part of Whatcom County, it is snowing. How about you, David? Is it snowing there in Seattle?
Yeah, we're getting a good amount of snow. It's pretty.
Nice. It is pretty. It's nice to see it. A good day to be cozied up learning about the octopus. So yeah, great. I was just saying to David earlier that I always think of octopus as tropical animals, but a lot of his work is done up in Friday Harbor on San Juan Island. So I guess we'll learn today, but they are not just tropical beings. They also love cold water. So a little, just welcome everyone. And while people are settling in, I just want to begin with a little housekeeping. So David is going to give his presentation, and we will be taking Q&A. You can write them in at the bottom there. If you look if you look at the bottom of your screen, you'll see a Q&A part, and you're welcome to type any questions in there. And we also have Neil McKay here as our technology expert, and so he will be helping. If you have a technical issue, please just type it into chat, and he will try to help you with that. So today's program is a part of Whatcom Reads, and it's presented by the Bellingham Public Library, the Whatcom County Library System, and the academic libraries of Bellingham Technical College, the Northwest Indian College, Western Washington University, Whatcom Community College, and Village Books is our community partner. This year, we're grateful for financial support from the Equity Fund of the Whatcom Community Foundation, Friends of the Bellingham Library, Whatcom County Library Foundation, Whatcom County Library System, the Bellingham Public Library, Western Washington University, and Bellingham Technical College Foundation. Whatcom Reads is generously supported by numerous community collaborators. So working together for over a decade, these organizations are achieving the vision of an annual local literary happening that brings neighbors together to discuss books and the joy of reading. This year's featured Whatcom Reads title is Washington Black by S.E. Edgecombe. I encourage you to check out other upcoming events this year inspired by themes in Washington Black, and don't miss the March 4th to 5th online presentations with the author. Learn more and register for the events at our website, whatcomreads.org. Neil will probably put that in the chat so you can see the website for that. Before I introduce our presenter, I have one final request. Please fill out the evaluation. We read every one of them, and we use this feedback to improve Whatcom Reads and apply for grants. You can find the evaluation form at whatcomreads.org/feedback. Thank you in advance for letting us know what you think. So we are thrilled to have with us today Dr. David Gire, a neuroscientist from the University of Washington. Dr. Gire is the principal investigator of the octopus research at the Friday Harbor Labs on San Juan Island. His research involves exploration into the neural network of octopus, and he has presented research on numerous topics. The one that I was intrigued with was how octopus arms make decisions. So welcome, David, and we look forward to your presentation.
Great. Thank you for having me as part of this fascinating series, and thanks again for your support in this talk, Anne and Neil. So I'll get started talking about what I agree with one of the quotes from the book, maybe the gods of the sea. So we'll get into a discussion of a couple main, just absolutely amazing abilities of these animals, and those involve both the skin and their intelligence. So now, due to the limitations of Zoom technology, what I'm going to be doing with these videos, I understand they'll come through choppy for a lot of people. So I'm going to pause and point something out, and then I'll resume the video. It'll probably be a bit choppy for you guys as it goes, and then I'll pause again and point something out. So it's going to be kind of a series of images that come through choppy or less choppy depending on your bandwidth. But the idea is to at least get some video and some imagery that you guys can look at too. So what you see here is, this is a video from Keishu Asada, who is a scientific diver who lives in Okinawa. And what he works with the Okinawa Institute for Science and Technology, or OIST. And what he's doing here is he's diving off a coral reef, and he's filming one of the most remarkable octopuses in the world, in my opinion. This is called Octopus cyanea, or the day octopus. And what's so incredible about this octopus is that unlike most species of octopus, which typically would come forage or hunt at dawn, dusk, or during the night, so limited kind of visibility, this octopus is out in broad daylight, hence its name, the day octopus. And it needs to be able to hide from its the many predators that are trying to find it. So it has an absolutely spectacular camouflage display. So what we'll do here is watch Octopus cyanea try to blend into a coral reef. And there it is. And you can see just with a change of its musculature, it suddenly goes from blending very nicely into that coral reef to actually appearing in its kind of default coloring, which stands out quite dramatically from the reef. And there, back to camouflage. And just look at this. There's an octopus there. And that's what it would normally look like. So really an incredible ability to blend into its surroundings. And this is noted in the book. There's a great quote about the octopus, and what they say is that an animal that can change itself to match its surroundings just by contracting its skin. And indeed, that's what you saw there. Octopus's camouflage, not like if you think about the color changing lizards, for example, which use more of a hormonal system to slowly change color. Octopuses have precise muscular control of their body that actually enables them to blend in with their surroundings by controlling their skin. So I'm going to explain to you how that happens, and the smart skin of the animal, in that it uses a few different tricks to blend into an environment really precisely. So let's take a look at this. So on the left here, we have a model of an octopus. This model of the octopus is here so that we can start giving it the ability to camouflage. And we'll see as we add pieces of its camouflage system to the skin, we're going to see it gets better and better at camouflaging. And then we'll learn how they all work together to enable an animal to change, to blend in, camouflage with its surroundings simply by contracting its skin. So here's our octopus model. It's on the bottom of the ocean, a simulated bottom of the ocean here. And first of all, this is what the octopus would look like if all it could do was use a system called the chromatophore system in its skin. So chromatophores, if you look up at the top there, they're basically kind of ink-filled pouches, you can think of them as. And so the musculature around the pouch controls how contracted or expanded the pouch is, and that controls the color of the skin. So if animals only use the chromatophore system, what you would get is kind of a similar sort of blending ability that you see on the left there. And now what we're going to see is the chromatophore system in action. So this is here in the lab, we're imaging skin, and I believe in this case it may have been a squid that was being imaged, but you can see the sacs of the pigment, the chromatophores, and when I hit play here, they're going to start contracting and expanding. So there they go. So again, to pause just in case it's coming through too choppy for everybody, the idea here is that the muscles of the skin are controlling how wide these pouches are. So these pouches can contract down to a little dot, or they can expand out to these kind of larger, to this larger size here. And then depending upon, if you look at different regions of the skin, depending upon how expanded the chromatophores are, you're going to have a different hue of the skin in that point. So this animal could kind of paint its skin by using these chromatophores. Some people kind of think of them as like maybe pixels, but that's not completely accurate because you're not seeing color changing. You're just seeing the chromatophores becoming bigger or smaller. It's really just changing kind of the darkness of the different parts of the skin here, if you look at it from farther away. Okay, so that's what the chromatophore system looks like as it's working. Okay, this would be you know, acceptable camouflage maybe for a lot of animals, but the octopus is one of the tastiest creatures of the sea. It doesn't have any bones, so any animal that could capture it is basically getting, for its effort of capturing an octopus, would be getting like the perfect meal in the ocean, because everything there is going to contribute to energy. It's just muscle and nerve, basically. So with all these animals hunting it, it needs a much better camouflage system than what we see here. So now we get into something where the octopus can approach a much more accurate representation of its surroundings and blend in much better. So let's see how that happens. What you're looking at here is an illustration from Science Friday showing the 3 different layers of the octopus skin. The chromatophore layer at the top is what we were just talking about. And these are those pigment sacs that can contract and expand. And now the additional colors can be achieved to really blend into the background quite well if we start considering the iridophore layer and the leucophore layer. So 2 different layers of the skin under the chromatophores. So let's start looking at light coming into the system. So we know the chromatophores, we have light coming in, the pigments in the sacs can change the light that gets reflected back out. That's how camouflage in the chromatophore system works. But there are other things going on here. So here you see the chromatophores contracting, expanding, but then out here we can see some coloration. So light is hitting some substance in the skin and being the colors basically being shifted there. So what is this? Well, this is the iridophore layer. So light comes in, it can, depending on the angle of incidence against the iridophores, it's going to change the color that comes off the iridophores. So you can see same blue light coming in at different angles to the skin is now reflecting out as a different color. Okay, so then what do we have as kind of our final piece to the puzzle here to create this near perfect camouflage of the octopus? It's the leucophore layer. And this might seem kind of strange that this will help with camouflage. We're not changing the color at all here. What's happening is light's just coming into the leucophores and being reflected back out. Same wavelength. Now, if we think about it a little bit deeper here, you can imagine why this might be useful. If the environment of the animal has a lot of kind of different wavelengths of light kind of bouncing around, it might be useful to just reflect back a bunch of that light, so you kind of would just look like your surroundings just passively. And so the leucophores would let the animal do that, and it does not have to control the leucophores. Now let's think about what happens if we have all of this happening at once. So you have a whole bunch of different wavelengths of light. Here we're showing mostly blue light coming in at different angles and some green light. And then that's going to reflect back out. Some of these components of the skin change the color of the light. Others are reflecting exactly the same light that came in, just right back out. Okay, so that ability of the animal is what would get us to the point you see on the left. So you definitely can see there's an octopus there. Still not perfect camouflage. So to get to the truly remarkable camouflage that I think was so well described in the book, you need to have another component to the system. And that component is muscular control of the texture of the skin. And that's, we can see here, this is a Pacific red octopus from our lab. We work with a local species, Pacific red octopus and the giant Pacific octopus. This enables us to locally go out, capture the animals, perform behavioral experiments with them, and then put them back in the water. So what you see here is one of our animals showing off its papillae. And these are these kinds of extension of the skin here, which are very precisely controlled by these animals. So they can basically send motor commands out to the papillae, and that changes the texture of their skin. And this is showing how that's accomplished mechanically. So what if we add the papillae to the picture? Then we get to this. So let's go back because I know it's we're probably a little choppy on Zoom. So here we have an animal that is not texturing its skin. It's simply changing the color of its skin using the chromatophore system and the iridophores and reflecting out some light with its leucophores. But now if we give the animal the ability to change the texture of its skin, it suddenly has this look. And now we can see it's actually somewhat difficult to see the animal now. So this is how octopuses get to be kind of the champions of camouflage. It's a combination of texturing the skin as well as the three main components that are within the skin that I just described. So here we're going to see another video, and I'll pause again as we go. Just turn off music for now. Okay, so pause. Try to find the octopus in this picture. May be very challenging depending upon the quality of your video feed, but we'll start it again. We'll keep going. Okay, try again. Can people see the octopus? Just kind of think to yourself, do you see an octopus here? And one more time. It might start becoming obvious because Keishu is rotating around the octopus, but oops, let's go back. Let's make sure let's head back to where Keishu is trying to find that octopus. There we go. Okay, so now I'm going to play it through. All right, so he's rotating about this piece of coral, and that is the day octopus right there. And now I'll pause for a second, and now just, okay, I can definitely see that people have questions that are coming up. That's great. I'm going to get to the questions at the end so that we can just keep going through the content right now. So what we see right here is everything we were just talking about. We can see different colors of the skin, which is going to be caused by the chromatophores contracting, expanding. And then we see dramatic use of the papillae to create texture. So the animal's using the papillae, creating this texture, and that's giving it this ability to really utilize all these aspects of its skin to blend in with its environment. All right, there it is. And then finally, it's going to change back to its kind of normal coloration and escape. And now it's hiding again, changes back, and swims away. Okay. So how was it that it was able to combine all of the components of its skin to be so well camouflaged? Well, this brings out some work that's going on across the country. So over in Woods Hole, Massachusetts, there's a team of researchers who asked that question. They asked, how are animals able to mimic their environment so well? How, in this case, they used cuttlefish, which have a similar camouflage system. So how does that cuttlefish there look at its environment and decide how to change, and what are the limitations of its ability to change? So could a cephalopod mimic words, for example, onto its skin? It's the type of question that they're looking at here. So here we can see how these cephalopods would blend into their environments. You see there's a sand, a small pebble environment, then the larger pebbles on the right. You can see the animal, by using these patterns on its skin, is able to blend nearly perfectly into those natural environments. And now here's the same animal trying to blend in to artificial environments. Now I hope what you notice here is that it's kind of not great. So it's not like the cephalopods can look at an environment and then suddenly they change. So we were watching Finding Dory with our 5-year-old the other day, and there's an octopus in that one named Hank, who can blend in perfectly with everything. Like he goes in front of a tiled counter or something and he just blends in perfectly. That's far beyond the capability of an octopus, because these are evolved creatures. So the camouflage system of the octopus has evolved to solve a problem, which is hiding in its natural environment. It didn't evolve to solve the problem of perfectly mimicking its natural environment. And so all of these mechanisms that I spoke about how the skin is doing this, a lot of them have evolved right into that environment. So the way the iridophores reflect the light and change the light based on wavelength or based on angle, the way the leucophores are going to reflect light, all of that is finely tuned into the animal's environment in a way that makes it look like it can nearly perfectly camouflage in the environment, but it's not arbitrary camouflage. It can't just go, walking out in the city and then blend in with the skyscrapers or something. So we can see that quite clearly here. Artificial environments are going to be pretty difficult for an animal to camouflage within. So what does this mean for the octopus? Well, it sees the environment. It's going to process that information in its brain, and then it's going to control this chromatophore system with its nerves to try to mimic the environment. But importantly, it's mimicking kind of natural statistics of that environment. It's not taking a picture of the environment and putting it on its skin. So next we're going to, I'm going to show you some information that is just coming out from some local labs in the Pacific Northwest that's trying to get at how octopuses look at their environment and see it in order to be able to camouflage so well. So what you see here is this is a micro CT scan of an octopus brain and eyes, basically, from Friday Harbor Labs. And so this is the lens of the eye here and over here. And then this is a piece of the brain called the optic lobes. This is, and then this is the retina, kind of right next to it. So those are the optic lobes. And then this is that pea-sized main brain that is mentioned in the book, and that's pretty accurate. The main brain of the octopus is absolutely tiny. Oh, and it's also wrapped around its esophagus. That's a nice fact that they have in the book as well. Okay, so here we have light coming in through the lens. It's being directed to the retina in the back here. And then the retina is going to send on the neural information to the optic lobes. Now, the interesting part about the octopus eye is that it's very similar to our own eye in that it's a camera-like eye, it's a lensed eye. And you can see how it focuses the light into the back and the retina here. One kind of interesting factoid of the octopus eye, they don't have a blind spot like we do. So our retinas are actually, if you looked at the way our retina is arranged, it's backwards. The light actually has to travel through all the neurons and cells to get to the photoreceptors. And then the electrical activity from the photoreceptor gets sent back out towards this kind of front end of the retina. And then finally, it all coalesces into a fiber tract that leaves the retina. And where it leaves gives us a blind spot. The octopus's eye is actually right way around. So light hits photoreceptors that are out here. And then the information can just be kind of sent right back out to the optic lobes in the brain without having to coalesce into axons that go through a blind spot. So octopus eye arguably has better features than our own. But what's very different about their eye is that the retina is super simple. So, and our retinas are just kind of enormously complex neural circuits. You can think of our retinas as just these enormous networks that are kind of performing state-of-the-art image compression. You could think of it at everything that we see. So already on your retina, your retina is already processing what you're looking at and changing it a lot before it sends it to your brain. But here, the retina doesn't do very much. It processes maybe a tiny bit, but it sends most of it. The information from the retina arrives in the brain in the optic lobe here. So you can think of the optic lobe and the retina of an octopus as kind of like our retina. So then in here is where all the action is happening in visual processing for an octopus, this optic lobe. And so the exciting development in Oregon is that some of the first neural imaging experiments are being done in the optic lobe. So this is from Chris Neal's lab, and the work's being done by Judith Pungor, who's a postdoctoral fellow in that lab. They've loaded these neurons with a dye that will glow when the neuron activates. So I'm just going to play it here. And what you're seeing here is the activity of the octopus optic lobe. And this is one of the few experiments where this has actually been imaged. They're using kind of a fancy system that allows them to optically section the tissue and actually see individual neurons here that kind of form these large masses that are activating. And then they can start making some hypotheses about how this piece of the brain will process the visual world around the animal to allow it to generate those kinds of camouflage patterns in response to the correct visual input. So I'm not going to go too far into that. It's just interesting directions in modern neuroscience, allowing us to start to understand how these brains work. Okay, so next, let's go to the next one. So once the animal's seen something in its environment it wants to mimic, it's going to send the command out to the skin, and that's what it looks like. So what you can see is the chromatophores are definitely active, but there's also, now you can see the papillae raising. So the skin is like this kind of wave of muscular activation upon which you see these little papillae kind of flickering. And so what does this do? Well, if the whole skin's moving, the change of texture actually engages all of these different capabilities of the skin to actually enhance the camouflage. So the animal sees something in its environment, and then it textures itself, and it controls the chromatophores. By doing that, what it's essentially doing is kind of in a roundabout way controlling the iridophores. Because remember, when light comes into the iridophores, the color that comes off the iridophore depends upon the angle at which the light hits the iridophore. So if you're an octopus and you change the angle of your iridophores relative to the incoming light, you're going to change your color. So the chromatophores aren't the only way to change the color of the skin by moving. By changing the texture of the skin, the animal changes the angle in the iridophores, and that also changes color of the skin. And that's really where I think a lot of the action is in octopus camouflage, is being able to change texture. And then as you change texture, you're actually changing color. Okay, so these two systems, and we can think of the octopus as being able to both change its chromatophores and through texturing, change iridophores, and then the leucophores you can think of as tiny mirrors. If you change the angle of the mirror, you're going to be changing what you're reflecting. And that also is going to contribute to camouflage. So simply by changing texture, the octopus engages all of these kind of passive capabilities of its skin to very precisely mimic the color of the environment around it. Okay, so another great quote from when they're describing the octopus in the book is, the octopus is the creature whose brain is wrapped about its throat. I really like the language there. It's very, the brain wrapping around the throat, and a brain that's no larger than a pea, but who is clever enough to play actual games. So next we're going to get into that brain that's no larger than a pea, and we'll get into some of the cleverness of the octopus. And we're going to learn another surprising way of how the octopus is so clever when its brain is so tiny. So what we see here is, this is another Octopus rubescens. In this case, it's from Rainer Gillette's lab at the University of Illinois. And they've released the octopus into this little tank, and the octopus is going to try to hunt a shrimp. So it can see this is, all being done in daylight, and we can see how an octopus might try to accomplish kind of a difficult game, which is catching one of the fastest prey species around. So it's going to have to move very quickly in order to very quickly and very precisely to capture the shrimp. You can see it watching, and there, it got it. And now note the papillae. Let's pause. Oh, there it goes. Yeah. So now it's using its papillae. It's changed its texture, and now it's nicely camouflaging with this marine environment meant to mimic its natural habitat. Okay, now in our lab, we decided to, let's pause for a second here. We decided to test the capability of the octopus to perform this type of prey capture behavior, but we did it with a twist. We released them at night into a completely enclosed environment that is only being illuminated by infrared light, which they cannot see. And so we can monitor the behavior of the animal in complete darkness using our infrared camera system. And then we decided to ask, how are the octopuses going to capture the shrimp now? So they can't see it, they can't direct that kind of pounce on the shrimp, but what are they going to do? So this was an example of something that really surprised us about these animals. So let's see how are they going to be able to get close enough to this extremely fast swimmer in order to capture it without the shrimp realizing it and getting away. So note, and again, due to our zoom constraints, I'll just point out the octopus is in the back here, coming out of its home cage, or its home den back there, and then there's a live shrimp right here, and the shrimp is kind of hanging out on these rocks here. Well, so note at this part of the prey capture behavior, the octopus has completely exited its den, but now it's doing something very different than octopuses that hunt in daylight. It's spread its arms out, almost as if, as we might do if we were to walk into, if a human were to walk into a completely dark room, probably do something similar where you'd spread your arms out and start feeling around the room. So here it's done the same thing. It spread its arms out. And now it's carefully kind of moving along. Oops, let's head back. All right, it is going to be successful, but let's hit play here. Okay, now I'm just not going to comment so that we don't lose our, the video doesn't jump. So see the octopus coming out, spread its arms out, and now it's navigating. Notice that it has now moved its arms around the shrimp, but hasn't touched the shrimp. And then finally it encloses the shrimp completely and settles onto the shrimp. And there the shrimp realized there was trouble, but it was too late. So in order to capture the shrimp in complete darkness, what the octopus did was it essentially just wrapped itself around the shrimp. So let's see if we can go back. Here we go. Okay, so it came out, moved its arms around the shrimp there. And the shrimp here, importantly, this shrimp doesn't know what's going on yet. This octopus has just completely surrounded it. And then it closes in and eats the shrimp. So what I want to point out here is how incredible this ability of the octopus is. So this octopus this is complete darkness. It knows there's prey there. It knows that there's a shrimp there, and it knows not to touch it, because if it touches the shrimp will get away. It's too fast. So what the octopus does is it carefully surrounds the shrimp in complete darkness. Remember, it cannot see what it's doing. Carefully surrounds the shrimp and then closes down on it. So what is it about this animal that makes it able to do that? How is it able to navigate so precisely around the shrimp, locate the shrimp, and then close right on it? So now we're going to discuss kind of the trick that the body uses to do that. So what you're looking at on the right is, let's see. What you're looking at on the right here is a schematic of the octopus nervous system, the complete thing. This was drawn by one of our graduate students in the lab, Dominic Civitelli. And here you can see the optic lobes at the top here. And then here's that P-shaped brain that's that's wrapped around the throat. And then down here we have the key to the whole thing. So everything here is the minority of the neurons. So most of the neurons actually live out in the arms of the animal. And so this is where this kind of incredible intelligence of the octopus is really engaged. And this is where their unique ability to move these 8 pseudo-independent arms, and even to navigate effectively in complete darkness. This is where all of that is happening. So 2/3 of the neurons are in their arms, and the brain here has only a tiny fraction of the neurons. The optic lobes, each optic lobe has more neurons than the brain, in the central brain. So let's learn about the way, so in a way you can think of these arms as kind of big brains for the octopus. It's not just an arm, it's actually doing a lot of its thinking in this kind of structure here. So let's cut it open, so to speak. We're going to use another micro CT scan and look at a cross section of this arm. Okay, so now we're looking inside the arm. On the left, you see the suckers. We have the outer sucker here, where the animal would be touching the substrate, and then there's an inner chamber here, which is precisely controlled by muscles. This is the muscle right here. And so the way the sucker works is the sucker touches a surface, and then the muscle contracts and creates larger space in this area here. And so then what's going to happen, you're going to create negative pressure and you're going to create suction. So as long as you can seal, as long as the octopus can seal onto something with the edges of its sucker, it can create suction through this mechanism, and each of these suckers can be independently controlled to create suction onto a surface. Little pro tip here for people who want to do octopus husbandry. This is a real limitation for them. So if you want to create an escape-proof octopus tank and you don't want to put a top on it, so you just want open air tank, the octopus can't get out, just put AstroTurf around the edge. Because with AstroTurf, they're not going to be able to form a seal. So if they can't form the seal onto a surface like AstroTurf, it doesn't matter if they're contracting the muscle here, it's just not going to create suction. So we actually do that in the lab, especially up at Friday Harbor, where a lot of our tanks are open to the air. We just put AstroTurf around it and the animal can't get out. Okay, so that's the sucker system. And then here is the kind of incredible set of neurons I was talking about. These are the ganglia, these bulges here. And then there's a tract of axons or projections from the neurons running behind the ganglia. And then everything you see here that's lighter, that's muscle. So let's learn about how this works. So those are ganglia. There's the axonal tract that runs behind the ganglia. And this is sensory input. So the suckers are like super fingertips in a way. They have the density of mechanoreceptors on the suckers is far greater than the density on your fingertip. So these suckers are able to do very fine tactile discrimination as they're searching surfaces. And that probably also helps them to form a seal. So you can imagine each of these suckers is trying to get in a good position where they're able to form a complete seal on the surface, and they can do that by kind of scanning across the surface. So they're very agile. They can position themselves on a surface and then create suction and suck onto the surface. And that's all going to be caused by the mechanoreceptors on the rim here. Interestingly, they also have chemoreceptors, so they smell with their suckers. Each of these suckers is kind of like its own little nose. So that information is also coming in to each of the ganglia. So each ganglion is associated with one sucker, and each ganglion is receiving all of that input. So the smell input, the taste input, and the touch input is all going into this little control center here. And then that goes to the brain or the rest of the nervous system. But the important thing here is that this little piece of the brain is essentially processing all of this information right at the sucker. And then this also sends information to other ganglia that are nearby. So finally, how does this work? So slowly expanding on the left, you can see kind of the network in the arms. So if this sucker were to touch something tasty, that's how the information would be conveyed to the rest of the animal. It would go out the arm, and then it can even skip the brain and go to the other arm. So there's actually a ring in the middle of the animal here that goes between all of the arms called the Brachial Crown, and that ring allows the arms to communicate with each other without going to the brain. So the arms can be doing something that the octopus is not aware of. That's very true. Okay, and why can they do that? It's because the octopus doesn't have a sensory motor loop that involves its brain. Or what do I mean by that? So imagine this was your arm and you touch something. That information would travel, obviously up your arm to your spinal cord, and then up the spinal cord into your brain, where your somatosensory cortex would process the information. It would then transmit that information to your motor cortex. Other structures like the basal ganglia will help coordinate the movement. And then all of that's going to be sent back to control the muscles in your arm. So in a human, this is a big loop that is where fine motor control is not going to happen without the brain. I mean, you can think of reflex loops in humans, but that's not fine motor control. That's, you know, if we think of what a reflex is, it's not going to be something where you sit down and reflexively play the piano or something. So what's happening with the octopus is far beyond a reflex, and the loop that involves that goes from touch to thinking to moving is actually in the arm. So that's what I'm showing right here. So touch to the ganglion to moving, each ganglion is locally controlling the muscles around it, not just the muscles of the sucker, but also the muscles of the arm that are local to that ganglion. So you can almost think of this as a bunch of little brains that are interacting with each other, and they're all contributing to local decisions about how to move the muscles that are around them. Very, very different than how we do it. And now you can imagine this whole system can work together. And if this arm touched something interesting, now the arm next to it could move to help this arm. And that could all happen without the information reaching the brain of the octopus. These also do send information to the brain like that. So a very rapid communication system from each of the ganglia into the brain does exist. But what we know from numbers, if we just count the number of axons coming from these ganglia and getting to the brain, it's tiny. It's a tiny, tiny fraction of the amount of neurons that are inside of these ganglia. So what's coming out of this is like a very abstracted message that gets to the brain. You know, it may be, we're almost certain the octopus does not, the central brain, that pea-sized structure of the octopus, we're almost certain that does not know where the arms are in any precise way. So the kind of ability to spatially coordinate between the arms and within these kind of extremely flexible appendages, all of that ability is given to the animal by the local interactions between the ganglia. The brain is not doing any of that. The brain is not doing any precise control of these arms. So the amazing dexterity of the octopus, you can think of the experiments of them opening twist top bottles, breaking open Legos, things like that. All of that amazing dexterity is from their arms figuring out how to do that behavior. The central brain is not controlling it the way I'm moving my fingers right now. That's not done by the central brain. So what does the central brain do? Near as we can tell, it coordinates kind of global motor patterns. So the brain might say, you know, go right to the animal, and then the animal figures out, the arms figure out how to climb over the rocks and how to go in that direction. Or the animal might say, like, go eat that shrimp, and then the body figures out how to get to the shrimp and acquire it without scaring it away. So there are motor commands that come from the brain, but these commands are simple and they're integrated with what's going on with the local ganglia. So there's not a clear and easy path for the brain to get out to the muscles and control them. So I'll just show you some examples of how we try to study this. I'm not really going to get too much into the science here. I'm definitely open to more questions from people, and I'm going to show you an opportunity we have this summer for people who get really interested, who may want to come up and do some experiments with us. So what you see on the right is this is a deep neural network that we use called Deep Lab Cut that is able to track the suckers. This is no human is tracking those right now. By using this this deep neural network to learn how to track an octopus arm, we can start reconstructing where these suckers are in space and how they coordinate with each other to find things like food. And in this case, the octopus just stuck its arm into a box that it can't see into. So the arm has to do it itself. The octopus can't see what's in here. And then by using this program to, let's see if we can, there we go. Okay, we'll play. By using this program, we can basically figure out where the arm goes and how these suckers are interacting with each other, because we're watching all the suckers at the same time and seeing how they interact. This is kind of an exciting time for octopus research, because the ability to actually do this kind of tracking of the animal, this really precise tracking of the whole body of the octopus, we're pretty much the first lab to do it using this these new techniques, and they work surprisingly well. So what we're thinking in the future is that we're going to be able to track a lot of what octopuses do, both in the wild and in artificial environments that we build, and then we can start asking some very precise questions about how their specialized nervous system enables them to do a variety of agile behaviors. We also work with modeling groups. This is the group of Eva Canso at USC. At University of Washington, we work with Josh Smith's group to do similar modeling. And what this does is allows us to model the biomechanics of the arms too. So if we plug in what we think these suckers are doing, and we plug it into a computer model, we can start coming up with solutions about how the whole system works together to create the intelligence of the octopus. And then this is an example of kind of a scanning technology that's been developed in the lab. This is are some sensors from Intel that we're using, where we can, and this is again up at Friday Harbor in the same kind of setup. We're kind of getting a partial 3D scan of this octopus as we're kind of moving we're just rotating around in the computer here to look at the kind of 3D point cloud that came out of scanning the octopus. So one of our hopes with a lot of this technology is that we're going to be able to kind of digitize the unique cognitive and behavioral function of the octopus, so that we can create three-dimensional models that can be interacted with by humans. And we can kind of bring people into an experience with an octopus in a virtual environment. There's a team at University of Washington who's been working on this with us. It's quite a challenge, but I think it's going to be useful, because I think being able to engage with this alternative form of intelligence is, it's an experience that's transformative for Washington Black in the book, and I think it's the kind of experience that too few humans actually get to have. And I think there's a way through modern technology to bring that experience out in a very accurate and visceral sense to a lot of people. Okay, so here we see our dive team. So just to kind of close out the talk and bring us back to the experience of Washington Black going out to capture an octopus. Here's one of our grad students. This is Dominic Civitelli. And I think the book, the treatment of octopus in the book does really balance the, as a marine biologist, the need to capture animals and study them. But you also have a desire not to harm the animals, because you realize how outstanding and incredible they are. And I think our lab, we're very lucky in where we do these experiments, because being in the Pacific Northwest, where these animals are native, and being right on the Puget Sound where they live, we can just go out, dive for them, bring them into the lab, work with them, especially up at Friday Harbor. It's very easy, bring them into Friday Harbor, work with them, and then we can bring them right back to the water. And so here's Dominic releasing one of our subjects. So that's a giant Pacific octopus. And in this next video, you'll see the octopus has returned to its environment. And again, I'll apologize if the zoom is choppy, but the octopus should be, it's kind of right in the middle there. It's just returned to its environment. And this is about 60 feet down near San Juan Island. And you can see it lives in a kelp, kind of this kelp area here, and you also can see as it settles. So here's that octopus again. This is the giant Pacific octopus. And what it's doing here is it's settled in among the plants. It can also see the rocks around here. So now it's going to engage its camouflage system. We can see it's moving. You can now see some of the skin is starting to mimic the ground, the little shells along the bottom of the ocean there. And then you can see, I'll pause for just a second. You can see the papillae have raised up here. Just check on, okay. And now it's settled and it's back in its environment and blends in quite well. All right. So I'd like to thank everyone for showing up to the talk today. I think we hit exactly our hour. I'm going to be available for a while now to answer questions. So I'm going to go to the chat and the Q&A, and I'll start going one by one through the questions. As we're finishing, I'd like to thank all of the students who are running the projects in the lab, the organizations, Friday Harbor, the Institute for Neuroengineering, and the Center for Neurotechnology at the University of Washington, the Behavior and Benthic Ecology Lab at Alaska Pacific University, who collaborates with us, especially with the giant Pacific octopuses, our collaborators within the US and around the world. And then finally, I'd like to invite anyone interested to apply for a course that we're teaching up at Friday Harbor Labs this summer. This is, of course, going to be determined by public health. We were going to teach this in 2020. It didn't happen for obvious reasons. This year, I think between vaccinations and the really nice model they have at Friday Harbor on how to run science without excessive risk, I think that they'll try to run through most of the courses this summer. So I would encourage people interested to look up From Behavior to Robotics, Reverse Engineering the Octopus, which we'll be doing at Friday Harbor Labs July to August in this summer. The way they typically run things with COVID protocols is they'll have what I call a research bubble. So you would show up, bubble there, and then you basically can run your research in classes. And we did this last summer with our research, and nobody had any, we had no positives. So I think it's a safe way to do it, but a lot will depend on travel. So if you're interested, feel free to follow up with me, or you can head over to the course description at Friday Harbor Labs and apply through there. And let's do the last slide here. And then this is just some images from the course. So with that, I'll start answering questions. I'm going to head over to, first we'll check chat. Oh, great. Thanks, Neil, for posting the course description. And it looks like more snow. Okay. So now I'm going to head over to the questions. Let's see what we have. Okay. Great. So I'm going to see if I can organize these in a way that's okay. There's two questions right away on the shrimp, so I'm going to go answer live here. So one question is, how does the octopus know a shrimp is there in total darkness? And the next question is, how did the octopus know the shrimp was there? So this is something we're studying in the lab, like as we speak. Unfortunately, we had to call off our dive today for for obvious reasons. Not safe to be out in the water with the snow, but we're going to be diving for more animals to run through some behavioral experiments over the next couple months to answer those questions. We've been doing work that shows that the octopus can simply kind of throw its arms out into the water like you saw it doing. And when it does that, it smells the water. So it's actually smelling the water with its arms, and that's probably how it knows the shrimp's somewhere nearby. We've also found that they can, again, in total darkness, if we just put a little piece of food into a tank and we have some water flowing over it, they can actually navigate to that water flow and then just start moving towards the food, smelling the whole way with their arms. So they almost look like a moth going, or a butterfly going to a flower, moth going to a flower, or something like that. But they're actually using their arms the whole way to find things in the dark by smelling. We also think, and this is something that's just completely unknown about them right now, we think they do something called hydrodynamic imaging with their arms. Again, that hasn't been worked super well out, but in other marine species, they can actually sense the flow of water and kind of create an image of what's around them. Weakly electric fish are one example of animals that do something like that, where they actually kind of scan their environments. So the octopus we believe has a system that allows it to do something like that. So it can probably both sense flow and movement in the water, so the little respiration of the shrimp, the little twitches the shrimp was doing, and can also smell it. In one book I read, the caregiver claimed the octopus recognized him. Is this possible? That's another great question. That's one of the big ones we wonder about in the lab. One of our grad students, Lisa Marie, she was working with some octopuses to train them to recognize her for feeding. So she would be the only one feeding them, and these specific octopuses, and she was able to train them to do things like present, like make an artificial movement that they normally would never do. In this case, they would go up to the surface and invert. So they'd invert and open their arms to ask for food, and they would do that when they saw her. So I think it was a kind of a clever way she had of showing that they actually recognized her, unlike some of the anecdotal evidence where people say like the octopus just started acting like excited or different when they came in the room. In this case, she made a specific behavior that they could only do to her to get food. If they did it to somebody else, the other people would not feed them. So it does look like they can recognize individuals. But what they're using to recognize the individuals is unknown. I think some people even would say that they recognize faces. I don't know yet. Okay, do squid share any of these characteristics? That's a really great question. So I'll click on answer live on that one too. They share the chromatophore system and the iridophores so that they can, so color displays by squid are super dramatic. They almost look like they're flashing lights at each other. But squids use this system very differently than octopuses. So squid, if you think of like the Humboldt squid, which is a terrifying creature that forms these huge schools that go up and down the California coast, eating everything. These are like 5 or 6 foot long squid. They flash colors at each other to communicate kind of social standing, and that's different. Some octopuses do try to change color. The darker the octopus, the more dominant in certain interactions. So cephalopods do seem to use color change as a social communication to avoid fighting each other, but it's much more developed in squid. And then squid don't need to camouflage because they typically won't be at the bottom of the ocean, or they're not benthic. That meaning they're living on the bottom. They're on the open water, in the open water column, and they're going to be there's going to be less substrate to blend into. So they don't really need to camouflage, but they do need to communicate. And so it seems like these colored displays are used mainly for communication with the squid. Another little factoid, squid have tentacles. Those are these specialized arm-like appendages that have kind of that larger flattened part at the end that they kind of shoot out to grab things. Octopuses do not have tentacles. Octopuses have arms. Okay, yeah, did it locate the shrimp by smell? Yes, it did. How long can an octopus hold a given camouflage? Oh, that's a really good question. So short answer is I've never done that experiment, and I haven't read a paper where they've pushed that to a limit. The long answer there has to do more with metabolism. So it'd be like, I guess the question, it'd be similar to the question of like, how far can a person run? It's probably going to depend on, and the reason I put it that way is the skin system of the octopus is under a lot of muscular control. And I was just having a chat with a researcher named Kurt Onthank, who's out at Walla Walla, and he also is the director of the Rosario Marine Lab. He was saying that he thinks the skin actually may consume a huge portion of the octopus's metabolism. So if you think of a person running a marathon, you know, their legs are using a lot of energy. The octopus trying to engage in that really dynamic camouflage, like that day octopus that we saw, that might be similar to like a human, you know, running. So it's going to get tired. But whether the muscles would tire in the same way that a human's muscles would tire, not sure if it would be quite as dramatic, but they would need to consume a lot of energy to maintain camouflage. So I would say a lot will depend on how well fed they are, and if they, you know, how long they need to, if they're in an environment where they need to stay camouflaged. But it is an active, a lot of the camouflage to be effective is kind of actively undertaken. So it is actually muscles, you know, using up calories and being engaged. If one takes an octopus from the Pacific to the Mediterranean, will it adapt to the new fauna, flora, and its camouflage? Another good question. I would say that really speaks to this evolutionary riddle of the octopus. So there's one great riddle to the octopus. Well, many great riddles, let's say, but one about this topic. That is, octopuses are colorblind. So at least as far as we know, with the type of photoreceptors they have, they're not going to be able to see in the types of color that they camouflage in. So if that's the case, how do they do it? How are they able to camouflage in colors that they can't see? So one argument there is that they just evolved to do that. So you can imagine that over, you know, the millions and millions of years, hundreds of millions of years of evolution, going from the ancient nautilus into nautilus-like creatures into the modern octopus, which doesn't have a shell and needs to really effectively hide because everything wants to eat it. That long evolution, it could be that the animals that just wouldn't camouflage that well would be eaten, and so they'd be out of the gene pool, and so eventually you'd end up optimizing towards an animal that camouflages super well with the environment in which it lives, simply because you know, it's basically being selected for. So it could be that they have all these mechanisms, but those mechanisms are going to be finely tuned in towards the environment into which they evolved. If that is the case, then if you took, say, the day octopus and moved it to the Puget Sound, where the color spectrum is different, we would predict that it might have some issues blending in, as well as some of the native species here. I think you'd see that if we could take one, if we could, you know, in a perfect world, I guess just simulate the tropical environment for our cold water species up here. My guess is they would be absolutely horrible at camouflaging to like the coral reef around in Okinawa, for example. I think that would, like if you took a giant Pacific octopus and brought it to Japan and you wanted to see if it could camouflage on a coral reef, I think it would just fail. And that's just evolutionary pressure in the Pacific Northwest is towards the, as you saw at the end there, the very green shifted kind of color, and the visibility is not great. So the animals don't have to be, great at matching all these colors. And I would guess that they would not be able to. So I'd say it's a long way of saying I think that they're optimized into their environment. Some of them will be able to adapt a little bit just because environments aren't that different from each other. But I think that there's a lot of evolutionary optimization that happened. Okay, does the octopus go out in search of food, or does it just act when it comes upon prey randomly? We think they search. There's the group, David Shields' lab, who we work with from Alaska Pacific University. He's, I think, one of the only researchers who's actually done this, where he put radio tags onto giant Pacific octopuses to see what they do out in the waters off Alaska. And it turns out they behave a lot like mammals in some ways. So they go out foraging, they go out on these long kind of foraging trips at night, and they actually go searching. So they go out searching for food, and then they return to their den. So they have a den that they stay in all the time. They go out, search for food. They eat a lot of their food in their den. You can tell that because they're super messy. So the middens around giant Pacific octopus dens are just like, it looks a little bit like my kid's room. So they're really really messy. So that means they're eating in the den, and they're throwing out like the crab shells and the clam shells, and just right in the front of their den. So what I think they do is they go out, they search, they try to capture things, and then when they capture it, they can actually drag it back to the den, and then consume it in the den, and or even on the way back to the den, and then drop it. Mm-hmm. Let's see. How long do they keep the octopuses at the lab before releasing them? At Friday Harbor, we usually have a summer season where we do research when it's really nice up there, and it's a really great place to be. So typically we'll keep, like the octopus you saw released in the video, the Alpha Answer Live, they were up there, I think, for like 2 or 3 months over the summer, and then they were released at the end of the summer season. So typically we're cycling them in every few months and then releasing them. Okay, how far north is their range? Yeah, as I kind of alluded to with the Alaska Pacific University question, they go pretty far north. They're all the way up to Alaska. So yeah, very cold. The Interoctopus delphini, the scientific name of the giant Pacific octopus, they go pretty far north. So they're kind of an interesting species in that they're huge, and they can really grow to just enormous sizes. And they evolved in the deep ocean. So yeah, it's their color, their ability to camouflage, some of their behaviors were probably optimized down, thousands of feet in the deep, where humans, don't reach. So yeah, it's an interesting animal. Okay, so I'll just, do they teach the progeny? That gets at another quote from the book about how strange it is that they're so smart, but they're so short-lived. They do learn by observation, but we don't observe teaching. So if an octopus solves a problem, another octopus can be watching it do that, and it can kind of mimic it and do something similar to what it was doing. But the one that is being mimicked doesn't turn around and try to instruct the octopus that is watching it. So they learn, they learn from each other, but it doesn't appear yet that they teach each other. Okay, I think we probably hit. Okay, so there's a couple questions. Let me just see if there's anything. Oh, how does the texture make it all gray like that? Yeah, so some of I'm not sure what that was referring to. Okay, what is octopus nose? Okay, I think we've answered most of the questions. Let's go to done. Okay. Yeah, that looks like I've answered most of them in the question. I'll answer a few in the chat that I see here. Okay, they do search in both day and night, longer trips at night. Yeah, if you read some of Jennifer Mather's stuff, you can see some giant Pacific octopus behavior. Let's see. Thanks for speaking. Can learning more about octopuses have applications such as biotech? Yeah, we hope so. That's one of our goals is to model how the octopus moves so that we could use it to understand better ways to control things like soft robotics. Let's see, and okay, it looks like a lot of this is tech support. Okay, so I wanted to thank everybody. I think I've answered most of the questions, and if you see something that I didn't get to, I'd be happy to answer it.
Thank you, David. Thank you so much. This was amazing. And I think we learned so much. And now I don't think I'm going to go into maybe octopus husbandries yet, but I am intrigued about the course up in Friday Harbor. And as you said, that's the time of year to be up there. So totally
cool.
Fascinating. And yeah, they're amazing all about octopus. And we really appreciate your expertise, and so nice to have a local expert in our presence. And it was great that you read the book too. So I hope everyone else here who's been on this session today gets to read Washington Black if you haven't already. And don't forget to fill out our evaluations, because that does help us continue with Whatcom Reads and all these great programs. And yeah, David, thank you so much. Totally
Great.
educational.
Yeah, thank you. Yeah. Yes, it was really enjoyable. And yeah, thanks everyone for the great questions.